Good morning, everyone. So thank you very much to both Tom and Kathy, and of course to Paul Allen and the Foundation. We're incredibly grateful for the support for some of the research we're doing in the lab. And what I'd like to do today is tell you a little bit about CRISPR gene editing, where it came from our perspective, where it's going in the future, and some of the work we're doing sponsored by the Allen Foundation. So the story of CRISPR really started with a curiosity-driven project that was aimed at figuring out how bacteria fight viral infection. And it was really the direction of this project and the work that we did with collaborators that led to an understanding of this system at a mechanistic level, enabling it to be harnessed for a very different purpose, namely for gene editing. So the first time that I heard the acronym CRISPR was actually from my colleague at Berkeley, Jill Banfield, who is a metagenomics expert, and what she found was that a lot of bacteria have in their genomes a repetitive DNA element that has integrated into it, among a series of repeats, sequences that are derived from viruses. And these had come to be called CRISPRs back in the mid-2000s. At the time, nobody knew what they did, but they were found in about 50% of sequenced bacteria. So it was a sort of an interesting mystery to figure out why bacteria might be able to store viral sequences in the genome and carry those sequences through many, many generations in the context of this repetitive array, the CRISPR. And so we got interested in this because of my longstanding interest in the way that RNA molecules control expression of genetic information, and wondering about whether these CRISPR arrays might in fact be transcribed and the viral sequences utilized to protect cells from viral infection using an RNA-based interrogation mechanism. And so through a wonderful collaboration with Emmanuel Charpentier, we actually used biochemistry to figure out the function of one of the central proteins that makes up these CRISPR-based immune systems in bacteria, an enzyme called Cas9. This protein turns out to be an RNA-guided enzyme that has the amazing ability to recognize double-stranded DNA sequences that match a sequence of 20 nucleotides that occur in a CRISPR-derived RNA molecule. And when that match occurs, the DNA sequence opens up inside the Cas9 protein, allowing the enzyme to make a precise double-stranded break in the DNA. Now, in the course of this biochemical investigation that we were doing, we figured out that in nature, this enzyme requires not just one RNA molecule, this one here, that comes from the CRISPR array, but also a second RNA called tracer that forms a structure enabling assembly with Cas9. So it's a dual RNA-guided DNA cleaver. And furthermore, it's programmable. So it can be programmed to recognize essentially any DNA sequence by changing the sequence of the RNA in this position right here. Now, Martin Jinek, a former postdoc in my lab who was doing this research, realized that he could simplify the system relative to what nature had done by linking together the two RNA molecules to create a single guide form of the RNA, enabling programming of Cas9 by simply changing this part of the RNA molecule right here and maintaining the same sort of handle structure on the other end. And for us, I think it was really that realization and demonstrating that this two-component system was effective at cutting effectively any DNA sequence that was programmed by changing the RNA molecule on this end, that made us realize that this could be harnessed for a very different purpose, namely for gene editing. Why is that? Well, that's because of a lot of the research going on over the last few decades that had led many labs to recognize that in animal and plant cells, the cells have a sophisticated machinery for detecting double-stranded breaks that occur in the genome and repairing them by pathways that involve either non-homologous end joining, in which a small change to the DNA can be introduced during repair, or by homology-directed repair, in which a DNA template can be recombined into the broken DNA, thereby integrating new genetic information at the site of the break. And so many scientists had recognized that if you could figure out how to introduce a double-stranded DNA break at a desired place in a genome, you could tap into these pathways and affect very precise alterations to the genome of a cell. And so what the Cas9 system does is to provide a tool for making those breaks in a programmable and readily harnessed way that can be implemented in effectively any kind of system. So I wanted to show you very briefly a movie that illustrates how we imagine this bacterial enzyme operates in a eukaryotic cell. And so what can be done is to put a nuclear localization signal onto the Cas9 protein, and that allows it to get into the nucleus of a eukaryotic cell where the DNA resides in the context of chromatin. And amazingly, somehow this bacterial enzyme is able to search through the DNA, here shown packaged with nucleosomes, and find a site in the genome that matches the sequence of the 20 nucleotide guide RNA. And when that match occurs, this protein with its guide RNA grabs the DNA and unwinds it and creates a double-stranded DNA break at a precise position, handing those ends off then to the machinery of the cell that allows repair to fix the break, sometimes by integrating a new piece of information during that process. And so this technology, which we published first in 2012, has now been adopted for a wide range of applications in many different kinds of cells and organisms and by labs around the world. So it's been a really exciting few years for us, sort of watching and playing a part in this technology as it's been rolled out for all kinds of applications involving alterations to the genome. So this is just a slide I put together that just shows many of the organisms that have been altered using CRISPR: plants, animals, insects, fungi, bacteria. And this is sort of opening a door to opportunities both in research but also with various kinds of applications. And I thought I would show you one slide that just illustrates a project that we've been working on now with colleagues at UC San Francisco, in which we're working on ways to deliver the Cas9 guide protein with its guide RNA into the brain to make precise changes to cells where eventually one could imagine clinical applications in neurodegenerative disease. And that's shown right here. So this is the work of a postdoc in my group, Brett Stahl, who came to the lab and was very interested in the opportunities for using Cas9 to edit the neurons in the brain. And so his idea has been to build on what we do in my lab, which is biochemistry, and generate purified protein molecules that are decorated with peptides that allow cell penetration. And so using those versions of Cas9, we can add guide RNAs that target this protein to particular places in the genome where we would like to make little alterations. And then we introduce these protein RNA complexes into the brain by injection to get across the blood-brain barrier. And we're doing these very precise stereotactic injections into particular areas of the brain, and doing this in a mouse model that has a reporter gene called TD tomato in the genome downstream of a series of stop codons. So by targeting Cas9 to the stop codon containing region, we can edit those out and turn on expression of TD tomato. And this is an example of some recent work from Brett where we're doing these injections and then taking sections from the mouse brain. And you can see that there's quite a decent volume of cells that get edited around the injection site when we do these kinds of experiments. So we're really excited about the opportunities to move forward using this kind of strategy for treating Huntington's disease as our first target, and then we'd like to eventually move towards working on Parkinson's disease, initially with animal models, and we hope eventually with our clinical colleagues moving towards human trials. So what else is out there? So this is really where the Allen Foundation comes in. So we've been working away with Jill Banfield, who studies bacteria and in their sort of native environments and tries to figure out how they grow, how they defend themselves, how they work symbiotically with other organisms. And one of the things that her research and others has turned up is that the vast majority of microbes on the planet have never been cultivated, and they've actually never been really studied in the laboratory either. And this is just a slide that illustrates this by showing you that just a small percentage of known microbes have isolated representatives, but the vast majority have no cultivated representatives. This is for bacteria, and up here this is for archaea. And so we are very interested in having a look at those organisms to try to find out something about their biology, with the idea that we're going to learn not only interesting fundamental biology, but also that there are going to be almost certainly a lot of interesting technologies that can be uncovered by understanding the mechanisms that these bugs are using to survive and to defend themselves and to thrive in interesting environmental niches. And this was really an idea that I started talking about with Tom Skalak, and then sort of developed into a project that involves Jill Banfield at Berkeley and some of our colleagues. And this has led to some recent work that we've done that started really with looking for things that are kind of like what we know is out there, but with some interesting twists. And this was really to ask, are there new forms of CRISPR pathways, these adaptive immune systems, in uncultivated microbes that we can find that have, because they have some features that we might expect to find in an adaptive immune system, but that look otherwise entirely different from any of the known CRISPR pathways. And this led to work done by 3 people in the group: David Burstein, a postdoc, and 2 graduate students, Lucas Harrington and Stephen Strutt, who teamed up together. David is a wonderful computational biologist, and these 2 students are experimentalists. And what they figured out was that they could search through these genomes, metagenomic data. These are bugs that, again, have never been actually cultivated. And they found examples of arrays that including sequences from phage, shown by these diamonds and boxes, that were next door to genes that encoded large proteins of unknown function. And they had some signatures of the kinds of things that we expect to find in adaptive immune systems, including a protein called Cas1 that is almost universally conserved in bugs that have a CRISPR array, because it's the integrase. It's the actual enzyme that grabs little pieces of viral DNA and inserts them into the array. So if you see Cas1 around, you kind of know that at least you've got a tool that's able to capture DNA and integrate it to form these kinds of repetitive sequences with newly inserted bits. So that led to the uncovering of an archaeal form of Cas9, which is the enzyme that was found initially in bacteria, had not been thought to occur anywhere in the archaeal domain of life, but in fact we found examples of this. And then 2 new enzymes, one called Cas-X and one called Cas-Y, that turn out to be proteins that are involved in RNA-guided protection of these bacteria. How do we know that? Because again, I told you these bacteria have never been cultivated. And so this is where the experimental work of these 2 students came in, and they realized that they could actually do an experiment, and I actually didn't think this would work initially, but they tried it anyway, thank goodness. And they figured out that they could transplant this entire locus, either for Cas-X or Cas-Y, into E. coli. And they had to do some things to make sure that they would still get good expression and make sure that these were under control of promoters that would be recognized in E. coli. But they were able to do an experiment that allowed them to test whether the presence of this locus in E. coli could protect the cells from the replication of a plasmid. And so this is basically just an experiment where we have a target sequence in the plasmid that matches a sequence in the CRISPR array of these associated proteins, either Cas-X or Cas-Y, and we can simply ask, if we have the target sequence in the plasmid and in the array, we call this a spacer, do we still see transformation with the plasmid? And the answer is we don't. And if we don't have a target sequence in the plasmid, or we use a non-targeting spacer that doesn't match any sequence on the plasmid, do we get transformation? And the answer is that we do. And you can see some of the actual raw data over here. And so this kind of system then allows us to, this is not biochemistry quite yet, right? But we're able to actually test the function of proteins like this very quickly, and we can make mutants and test their function in an assay like this that allows us to do experiments even though we are not cultivating the bug where these loci actually came from. So I just want to end by pointing out that gene editing is a really powerful technology, and it's being utilized in many different ways, including by various groups that are thinking about how to modify the human germline. And so, you know, are we right around the corner from CRISPR babies? I would say we're not. But nonetheless, we know that this technology is able to make targeted changes in the germline, including in the human germline. And so I've been very involved. One of the things I've been spending time on over the last few years is on encouraging responsible use of this and thinking about how we can work together as a global scientific community to try to ensure that this technology is used in a responsible fashion going forward, including being involved in this report that was released earlier this year from the National Academies. And I'd just like to close by acknowledging a terrific group of people that I have the pleasure to work with. And of course, all of this work on these new CRISPR systems and what else is out there is being sponsored by generous support from the Allen Frontiers Group. And we're very grateful for that. Thank you.
We have time for one question, if there's one.
Are you going after new enzymes in the metagenome or secondary metabolites, or what's the general target space for the metagenome?
Yeah, no, in the metagenome, we're very, very excited about other ways that these bugs are defending against bacteria. And in fact, there's some evidence that there are pathways that are not CRISPR, but that are probably involved in either antibiotic generation or some kind of antiviral defense that we're curious to investigate. And these are really, you know, probably going to be whole pathways of genes that are doing something that we don't understand the function of yet.
Here's a quick one. Is the archaeal Cas9, is that horizontal transfer, or is that actually archaic?
Yeah, it's not clear. It's a curious enzyme. It's quite a bit smaller than the bacterial Cas9s that have been identified, and we were never able to reconstitute activity, even though there's good evidence in the metagenomic data that it's part of an active CRISPR pathway that's acquiring new spacers. So presumably it's functional in that organism, but we haven't figured out actually how to really test it functionally yet and really where it came from.
Okay. Thank you so much.
Thanks.